Welcome friends, I am Dr. S.MEGALA, Assistant Professor, Department of Microbiology, St.Joseph College of Arts and Science, Autonomous, Cuddalore. Now I am going to talk about fermenter. Fermenter is used for commercial production in fermentation industries and is a device in which a substrate of low value is utilized by living cells or enzymes to generate a product of higher value. Fermenters are extensively used for food processing, fermentation, waste treatment, etc. Fermenter is a vessel that maintains optimum environment for the development of significant microorganisms used in large-scale fermentation process and the commercial productions of products like alcoholic beverages, enzymes, antibiotics, organic acids, etc. Fermentation means is an anaerobic pathway, a common pathway in the majority of prokaryotes and unicellular eukaryotes. In this process, glucose is partially oxidized to form acids and alcohol. In organisms like yeast, the pyruvic acids formed by partial oxidations of glucose is converted to ethanol and carbon dioxide. A fermenter should provide for the following: agitation, aerations, regulations of factors like temperature, pH, pressure, aeration, nutrient feeding, liquid level, etc. Sterilization and maintenance of sterility. Withdrawal of cells or medium. Next one, size of fermenter. The size of fermenter ranges from 1 to 2 liter laboratory fermenters to 5 lakh liters, or occasionally even more fermenters are up to 1.2 million liters have been used. The size of fermenter used depends on the process and how it is operated. Next one, construction of fermenter. Industrial fermenter can be divided into two major classes, anaerobic and aerobic. Anaerobic fermenter require little special equipment except for removal of heat generated during the fermentation process, whereas aerobic fermenter require much more elaborate equipment to ensure that mixing and adequate aerations are achieved. This one, image of fermenter. Next one, parts of the fermenter. First one, cooling jacket. Large scale industrial fermenters are almost always constructed of stainless steels. A fermenter is a large cylinder closed at the top and the bottom, and various pipes and valves are fitted into it. The fermenter is fitted externally with a cooling jacket through which steam or cooling water is run. Cooling jacket is necessary because sterilization of nutrient medium and removal of heat generated are obligatory for successful completion of the fermentation in the fermenter. For very large fermenter, insufficient heat transfer take place through the jacket and therefore internal coils are provided through which either steam or cooling water is run. Next one, aeration system. Aeration system is one of the most critical part of a fermenter. The agitator, stirrer glands and bearing baffles the aeration systems. The two separate aeration devices are used to ensure proper aeration in fermenter. These devices are sparger and impeller. The sparger is typically just a series of holes in a metal ring or nozzles through which filter sterilized air passes into the fermenter under high pressure. The air enters the fermenter as a series of tiny bubbles from which the oxygen passes by diffusion into the liquid culture medium. The impeller is an agitating device necessary for stirring of fermenter. Next one, baffles. The baffles are normally incorporated into fermenter of all sizes to prevent a vortex and to improve aeration efficiency. Next one, pH and temperature control. A system of pH and temperature control should be provided. Next one, inlet or sampling ports. The fermenter should be provided with inlet ports either manually or mechanically to allow the sterile inoculum. Necessary sampling ports are to be provided from middle of the fermenter to the bottom to collect the fermented liquid. Next one, drain point. The fermenter should be provided with a drain pipe to collect the fermented liquid for necessary processing. The drain point may be connected to other processing plants for final product recovery. Next one, peristatic pumps. Necessary pumps are to be provided for allowing the liquid or to take the liquid from the fermenter. Next one, filters. On-line filters are also to be provided to allow sterile air inside the fermenter. Next one, service provisions. The fermenter should be equipped with service provisions, compressed air, sterile air, cold water, hot water, steam with pressure, steam condenser, electricity, generator, motors, control or monitoring devices. Next one, fermenter and its control systems. The heart of industrial fermentation is fermenter. Fermenter is a type of the bioreactor. It is a system provided with controlled environmental conditions for the optimum growth of microbes in liquid culture and productions of specific metabolites. It is a device in which the microbes are cultivated and motivated to form the desired product. It is contaminant systems to provide the accurate environment for the optimum growth and metabolic activity of microbes. Fermenter prevent the entry and growth of contaminating microbes from outside. Thank you.